Welcome back to Tenure License. Today's topic is about the ovule or the ovary culture. So before going in detail, let me tell you about the parts of a flower. So we have already discussed in the anther culture that the androecium or the stamen consists of three parts. First one is, as you can see, anther, that is bilobe, and the filament, and the bilobe are connected by the connective tissue. Okay. So the next is the female reproductive part of flower, that is called the pistil. It consists of three parts. First one is the stigma, and this slender-like structure is the style, and this snake-like structure is the ovary. And inside the ovary, the ovules are present. So after the fertilization, the ovary becomes the fruit, and the ovule it becomes the seed. Okay. So ovary culture is a technique of culture of ovaries, obviously isolated either from pollinated or unpollinated flowers. Okay, so it developed by niche in 1951, and ovary. What is ovary? Ovary is a oval bearing region of a pistil. So this is completely the pistil. So what is ovary? Ovary is the region which bears the ovule. Okay. So for the non pollinating plants, the culture is we need to put the synthetic oxygen like uh, we have already discussed 240, right? So next is about the structure of the embryo sac. So the ovule, you can see the ovule is the embryo sac plus the integuments. Okay. So ovule is the embryo sac plus integuments. Okay. So these are the unfertilized cells of the embryo. First, three antipodal cells in the chala's gland, polar nuclei in the central cell, already mentioned, and in the micropolar end, there is one egg cell and synergids. That is completely called the egg apparatus. Okay. So then, what are the applications of the ovary culture? So it helps in the production of the haploid plants, and it achieves the in vitro fertilization. The next switch to the culture part. Okay, so just like the anther culture, here also two steps. One is direct method and one is indirect method. And the indirect method always follows the intervening sequence, that is the callus formation. So one is direct method, one is indirect method. Okay. So in the direct method, from the egg cell and synergids and the antipodal cells, they are the cells will organized and the cell divisions occur, leading to the formation of the pro-embryos. Okay. So in the direct stage, first the egg cell, the synergids, the antipodal cells organize and they undergo the cell division to produce pro-embryos. Okay, and then that will differentiate into the embryos. Then the differentiation occurs and there will be the formation of embryos. Okay, so this is the direct method. In case of indirect method, as I said, there will be formation of intervening callus. Okay, so in between the synergids, egg cell and the antipodal cell, and in between the embryo, there will be the formation of first the callus. Okay, So in the indirect stage, the callus will form, that is the intervening stage, and it can form directly either from the egg cell or from the signature or from the polar nuclei which is present in the central cell or from the antipodal cells. It may develop from the true embryos. So next step will the plants will get regenerated from the callus, and then it will produce the haploid or the diploid or the mixoploid plants. Okay, so the first successful ovule culture reported in barley by San Nguyen, and it is also successful in onion, that is Allium sativum. So this is all about the ovary culture. If you like this video, hit the like button, share with your friends, and do not forget to subscribe to the channel. Thank you.